ctDNA is a new technology, and there's a lot of different ways that we can use it. We can use it to give us information about the molecular characteristics of a tumor. We can also use it to tell us if there are potentially some cancer cells still in someone's body, right? So this idea of, finding a tiny fragment of tumor-associated DNA is pretty powerful, because you do surgery or chemo or some modality like radiation, you expect you're gonna have killed off all the cancer cells. And if there's a signal still there that there is tumor DNA floating around, depending on your assay, but with the newer generation ones, you have an overwhelming chance of having your cancer come back if nothing is done. So we have staging for a reason. Our staging, our guidelines, our radiography, CEA, whatever tool you might choose, they're all there to stand in for the fact that we don't have X-ray vision and we can't individually spot cancer cells in someone's body. And they give us this information of what is your recurrence risk? Well, now we have a new tool, and if you integrate that information, your recurrence risk, even if you have a low-risk stage 2 patient that's clean on scans with clean CA, goes from very low to almost certain. So to me, it's a signal that we probably should do something different. I'm really grateful that at Northwell we have clinical trials for patients that are ctDNA positive. And I would encourage people that if you have a patient and you're sending this assay and it does come back as ctDNA positive, if you haven't considered adjuvant chemotherapy, you probably should. And if you've already done that, you should probably start thinking about trials and other next steps.
One of the things I love most about this show is getting to introduce someone to an audience before the field fully catches up to them. My guest today is Nicholas Hornstein, MD, PhD from Columbia, fellowship at MD Anderson, and now building a clinical research program at Northwell Health around one of the most pressing questions in colorectal cancer right now. What do you do when a blood test finds cancer that no imaging can see, and you don't yet have a drug proven to clear it? What I find most compelling about him isn't just the science, it's the honesty. After his INTERCEPT-TT trial results, he said publicly, and I'm going to ask him today to say it again, that the field needs better drugs than it has right now. That's a remarkable thing to say about your own trial. It's also exactly the kind of thing this show exists to put on camera. This is the Moonlight Shift. Pull up a chair.
Gina, thanks for having me.
Dr. Hornstein, we are so happy to have you here. Welcome to the Moonlight Shift.
Pleasure.
Thank you so much. How are you doing? Congratulations on your new baby girl.
Thank you. I am sleeping a little bit, but it's great. It's a lot of fun having another little girl at home. So now I'm a permanent girl dad of 2.
I was just gonna say, what is life like as a girl dad, coming from a full-time boy mom?
It's different. It's awesome. I'm really looking forward to how they're going to grow together, but I'm learning new tricks. I'm a little worried about college and high school and all those things. There's other problems I'm going to have to deal with.
Fair.
I'll figure that out later. Yeah,
good.
Dating, no, never going to happen.
You're going to do great, one day at a time.
That's exactly right. One night at a time is really how I'm thinking about this, but yeah.
One night at a time, I like that much better. Now, obviously, new baby girl, sleep is #1 priority right now in terms of, and also taking care of your baby. But what does life look like for you outside of practice? How do you turn off, decompress from work?
It's a challenge, it's a struggle.
Yeah.
I think we talk a lot about work-life balance, but at the end of the day, being an oncologist means really being on call for your patients day, night, whenever it is. So it's a little tough to turn off. For me, I'm glad I have a really supportive family, and being able to just be with them, going to the park with my daughter, playgrounds, going clamming next week, right? So, you find things to enjoy and do together.
Do you have your go-to places you like to go eat as a family?
I cook a lot. So my wife does not cook.
Okay.
She has tried. She has set fire to the kitchen multiple times.
Okay, so we'll keep her out of the kitchen.
So that's on the outs. So I'm the one that ends up cooking, and as a result, probably 90% of our meals we cook at home. Yeah, because she doesn't have to do it.
No, that's great. What's your specialty? #1.
So, funny story. Long time ago, back before I decided to go to medical school and do all that, when I was in high school, I thought maybe I'd like to be a chef. I started taking classes in the culinary school, started, testing out being in a kitchen. And it turns out being in the kitchen's really, really hard. And also cooking for people you don't know kind of sucks. So between those two, I picked like a way easier route, which was, MD, PhD, oncology, that kind of stuff.
I love that. Would love to see, we should almost create like a collage of your home-cooked meals one day. I bet it would do great in a cookbook. Just saying.
If I ever have a cookbook, it'll probably be Greek. My mother's maiden name is Dimitidis, so growing up in a Greek kitchen, it's hard to escape that.
I love how naturally you just said that. Now, you mentioned the MD and PhD, so I want to talk about that.
Yeah.
You did both. Now, there's a moment in every MD, PhD training. You have to decide, are you primarily a scientist who sees patients or a clinician who does research? And where did you land on that? How does that degree really set you apart?
Yeah, so it's interesting you asked that. At a lot of different time points in my training, I was offered, hey, you can take a year off the clinical time, you can do more research, you can not cut this corner, but change the amount of time you're going to invest in clinical training to really push the research forward. And I always found that really a challenge to wrap my head around, because if a patient is coming to see me, I am their clinician. They don't care how many papers I've published or grants I've gotten or any of those types of accolades. They care, do you know how to treat me clinically? And I just, from my perspective, didn't think it was appropriate to cut that training down. It's there for a reason, probably, I hope. So I always weighed more towards, I'm gonna make sure I have the best clinical training possible. And at the same time, I'm gonna try to pursue this PhD. I'm gonna try to really hone in on my scientific chops, computer science, those kinds of things. Because at the end of the day, I really wanna do both. I like to think of it as being like pluripotent, right? I can do a lot of different things. There's a lot of different tools in my toolkit. And yes, it took more years, but in practice now, I'm seeing patients in clinic, but I'm also driving forward research initiatives. I'm using my background, computer science, I'm using my knowledge of next generation sequencing to really drive system initiatives forward. Because the reason I did a PhD as well, took more time in school, kind of gave up most of my 20s, as most physicians do, was because I thought there might be a way that I could help more patients than those I could just see in the clinic. And we can see a lot of patients in clinic for 4 or 5 days a week, but how do you have a broader impact? And to me, the answer to that question was, well, I can do research, I can help design the way we implement workflows, the way we're doing testing, giving information, whatever it might be. And that's what I've tried to do now in practice.
I think that's brilliant. I want to talk about your background and your journey to get here. So you grew up in LA, you had MD Anderson for fellowship, and now you're at Northwell. And I would love to hear your thoughts on what does it mean to take care of New York patients specifically? Yeah,
so one other thing to mention, I went to medical school here too. So I was at Columbia for the MD and the PhD. So I had my first taste of New York patients then. It's different, it's funny. So I was at residency at UCLA. So I have Los Angeles, I have New York, I have Texas. So like a little bit of the West Coast, East Coast in the middle. The patient population in each place is drastically different. And I think saying this out loud for people that are practicing there, they probably know this. There are aspects of each of these patient personas in each location, but they're more concentrated when you're actually there. So definitely New York is a melting pot. There is, the Upper East Side socialite. There are the kind of uninsured indigent patients, and there's everything in between. And it's really amazing to see that diversity. Texas, we had a lot of people that were coming in with different levels of medical literacy. We had some people flying in from out of the country, but we also had some people, driving in 6 hours from more rural locations. Yeah. So again, diversity, but in different ways. And then Los Angeles, a lot of discussions of alternative medicine, a lot of different approaches to care. And either way, it's just, the patients are different. The oncology is the same, but it's just really cool to be able to interact and see these different facets. And each region, I feel like has its personality come out in its patients.
Did you say when you were in Houston that people came in from out of the country?
So at Anderson, there was a lot of different international relationships that were developed over time. So patients would fly in, supported by their government, to come and get oncology care, which was really interesting.
Yeah, that's like a very unique opportunity. Very cool. Okay.
Lots of translators.
Yeah, I imagine. Yeah. So I want to talk about some of your trials. So first of all, how did you go from bench science at Columbia and MD Anderson to running a trial specifically designed around a liquid biopsy signal? What made that the problem that you wanted to solve?
Yeah, a lot of this stems from what I did at Anderson. And when I was there, I was doing a lot of work in the GI department in colorectal cancer. My background, my PhD, is in computational systems biology. And as part of that PhD, worked a lot with next generation sequencing. So really feel like I have some insights into, hey, these are new tools, new assays that we can apply to improve oncology and outcomes for our patients. So when I was at Anderson, I really was looking for ways, how do we integrate these technologies into clinical trials, both the design and the overall readout, so that we can get new things to patients faster, and also really use these technologies to their fullest. So there are a few different projects I have running now. Some are using liquid biopsies. A project that I worked on at Anderson used a multi-cancer detection test to try to identify cancers of unknown primary, which seems like it worked fairly well. I think the trial you're referring to was one that was looking at treating patients who were ctDNA positive on a minimal residual disease test, and then randomizing them to either a treatment which was Lonsurf, which is standard care for third-line metastatic colorectal cancer, or observation. Is that the one you're on?
Yes.
Because now there's a few more that...
we can talk about as many of them as you like.
That's good. That's enough. There's a lot of trials out there, right? So really the idea is we've got these tools. They are too slowly getting integrated into our guidelines. And how do we generate the evidence to help people make these assays actionable? Really say, okay, this is my result. How do I help a patient with this result? That's kind of what I'm trying to get at.
Well, you have also said before that we need better drugs than we have right now.
Yes. So, one of the challenges, I say Lonsurf, and Lonsurf for the right patient can be really helpful and, give them a few months of life back, but it's a few months. And we know we should be able to do better than that. So looking to this next generation of assays, both the tests and this next generation of drugs, I see that there's a lot of hope out there for the future in terms of, taking a patient that has a, over 95% likelihood to recur if they're ctDNA positive, and being able to change the natural history of that, which the natural history is you have a recurrence, maybe you have metastatic disease now. How can we avoid that? The answer probably isn't drugs like Lonsurf. The answer is probably drugs that are still in the pipeline, that are still in phase 2, 3 clinical trials that aren't approved yet, that have much better response rates. Remember, Lonsurf has a 0% response rate. Not that I'm knocking it, but that's the data. Mm-hmm. And now we have drugs coming out that are really showing significant activity in refractory colorectal cancer. If you can't guess, my focus is on below the belt malignancies, so colorectal, appendiceal, anal cancer. That's where I focus the majority of my time.
So I want to hear about your research program also that you're building. Let's talk about that. What has that whole journey been like for you?
Yeah, so coming to Northwell, it was a little bit of a different choice than some of the other institutions that I was thinking about joining after fellowship. And I saw it as this major opportunity where at the time there wasn't as a mature research program in the GI space. So Dan King was there. He's doing a lot of really amazing things in pancreas cancer, and then some other investigators. But coming in, it presented an opportunity where we're diagnosing 26,000 new cancer patients every year in the system. That's a huge amount. So if you can bring in, investigator-initiated studies, if you can bring in sponsored studies, if you can really help grow the back-end bioinformatics that we're using to identify patients to make sure we're giving the right care to the right patient at the right time, you can help a lot of patients. So coming in, have been able to really grow the program. We have a half a dozen investigator-initiated studies that I'm PI of or co-PI of. In colorectal cancer specifically, we're growing out the sponsored program, and at this point, we basically have a study for every patient with colorectal cancer wherever they are in their treatment journey, whether it's localized or metastatic, and that wasn't something we had 2 years ago. So I'm really proud of the work we've done as a team, because it's not me, it's like the team has done this. And I'm really looking towards the future and continuing to grow that across the GI portfolio and then potentially into other tumors as well.
It's so exciting.
Yeah.
It's a huge advantage, not just for the New York area, but I think it really sets a wonderful example of what other centers can also emulate.
Yeah, I think that a lot of this is based on industry partnership, which at the end of the day is actually really important to help make sure we're providing that type of care. Because look, we are in clinic, we are with our patients. People on the other side of things, whether that's in biotech or pharma, are helping to develop these treatments. We got to work together to figure out the best ways to get them to our patients and find the best patients for these treatments. So a lot of what I'm working on now is also leveraging my computational biology background. I'm a computer nerd at heart. I always have been.
Love it.
So we're building these enterprise level, I don't want to say AI systems, but large language models, natural language processing tools that are helping us move all of our data from these messy pathology reports. Okay, pathology reports aren't messy. Structured, beautiful pathology reports and radiology reports and notes, and putting them into a structured format. And we can then use that to identify patients for trials, to make sure patients are getting the right molecular sequencing done at the right time. And the goal is that we can really improve the care we're providing across the system.
I love that. Thank you so much for sharing all of that. I think it's great. We talked a bit before we started here on the Intercept TT. There are a couple of trials that are under there and one that has had some updated data within the last couple of days. Do you want to maybe talk a little bit about that program overall and what we just learned within the last couple of days?
Sure. So Intercept, just to introduce people to this concept, is a program at MD Anderson that is this idea that when patients have minimal residual or molecular residual disease in colorectal cancer, they're going to recur. Like, that's the natural history. It's going to happen if you give it enough time. And we probably don't want that to happen, because we know when a disease comes back, especially in its metastatic form, it's going to be much harder to cure that patient. So Intercept is the idea that we can intercept the cancer before we have a radiographic signal. So you have a positive signal on an assay, like ctDNA test. Let's stop that from ever showing up on a scan. So there's a number of trials that are ongoing now or have completed enrollment where they have been using different treatments or strategies to prevent that recurrence. There was one study in particular that I was part of. Andrew Pellett was the first author, as I recall, and Vin Morris was the last, that was looking at using Lonsurf, which we mentioned previously, to prevent that recurrence. And in patients in that smaller study, it did seem to prevent it. There's new data out of Japan as part of a very large ctDNA effort called ALTAIR, which showed that this strategy, although it delayed progression-free survival or disease-free survival by about 3 months, it didn't really change the course of the disease. So not a positive study. The p-value is like 0.11, not that p-values are the most important, but it goes back to this idea that we probably need better therapies to really change the disease course.
I think that makes sense. I think also, too, coming out of ASCO, we probably saw a lot of different types of newer generation therapies that are probably available.
Yeah.
I think it's always interesting to just kind of see the early signals in some of the phase 1 data, even if it's too early for a lot of people to start talking about. But was there anything that really kind of just caught your eye at ASCO that maybe a lot of people aren't saying?
Okay, so the first thing to mention, and everyone is probably saying this, daraxonrasib, pan-RAS inhibitor. RAS is driving 90% of pancreas cancers, 40% of colorectal cancers. Standing ovation, plenary session. In second-line pancreatic ductal adenocarcinoma, they're probably going to get an approval the moment the company actually sends in the application to the FDA. Huge game changer, yay. Okay, we covered that. Past that, there's a lot of other molecules that are coming out AbbVie, for example, has a c-Met inhibitor that has in all comers in the refractory, like fourth line metastatic colorectal setting, a 23% overall response rate, which is outstanding. But there's other drugs that are earlier in phase 1. We just saw in pancreas cancer a combination of a RAS inhibitor plus a MTAP deletion PRMT5 inhibitor got like an over 90% disease control rate in pancreas cancer. So that's a phase 1 trial. It was like 97%. But those numbers are incredible for pancreas. So there's a lot of things coming that are really exciting. There's lots of HER2-directed therapies, some of which are going to be standard of care soon that are coming. And I think the future is pretty bright now that we have better tools, and they're going to continue to evolve. We're going to continue to see antibody drug conjugates, molecular glues, all these newer tools really coming into the clinic after their phase 3 trials are completing this year, next year, and so on.
Thank you for highlighting those. I think that's great. My final question. So you've been quoted as saying you were drawn to hematology oncology because it's a good place to think about science and life on a grand scale. So sitting here today, what does that actually mean to you in the context of the work that you're doing?
It's a good question, right? One of the reasons I chose to focus on hem-onc is I feel like it is very rewarding to be able to be there with a patient on what is probably the worst day of their lives for one of them. And I think that you can make the case for that for a lot of fields, but especially in oncology, I think there is this human connection that's so essential. There's patients that we're treating that we know we're not going to be able to cure. And being there for them, providing that type of support, not just the knowledge of, hey, this is the next best treatment or this is how we should, go about managing this, is something very valuable to me. And it really makes me feel like I'm working on something important. And that's really what drives me. That's what drives me to be in clinic with my patients, what drives me to run studies, what drives me to help with health system initiatives. Because, your first question, your work-life balance, it's a lot of work. And a lot of it is, working at night or on weekends or times when, probably I should be hanging out with my family. So there probably should be some more balance, but I think there's a lot to do, and we only have so many years to do it. So I take it as a challenge, and it's something that motivates me and wakes me up in the morning to try to do better for the patients that I serve.
Well, I know I can speak for everybody here at MGH and definitely everyone at home that you are definitely doing something important. So thank you for all that you do. Yeah. Dr. Hornstein, are you ready to play a game?
I'm so ready to play a game. What are we playing?
The name of the game is liquid biopsy or crystal ball.
Oh boy, okay.
Get ready. So.
I have no idea where this is going.
We're gonna roll with it.
Yeah, let's see.
So there's gonna be a couple options. You spend your career figuring out what ctDNA can and can't tell us. So I'm gonna give you a scenario about liquid biopsy and you tell me, is this something ctDNA can actually do today, something it might do in the future, or something that's more crystal ball than science?
Okay, let's go.
Ready? One pick, one sentence, no lectures. Using a ctDNA test to decide whether a stage 2 colon cancer patient needs adjuvant chemotherapy.
It's here. Done.
Liquid biopsy can do this now. Is that your answer?
MRD testing can do this now. We have data from observational studies, Galaxy Japan, as well as cooperative group trials out of Australia and New Zealand. It certainly can help.
Okay. Long run-on sentence. I'll let you have that, though. That's good.
I was never that good at grammar in English.
A blood test that catches colorectal cancer recurrence before imaging can see it, and then you have a drug that actually clears it.
Future. We're getting there. Clearance is tough. Not there yet.
Fair. All right. Using ctDNA signal to select which patients in a clinical trial actually need to be enrolled, adaptive trial design based on liquid biopsy.
These trials are already running, and many more are coming. In fact, the only problem with these trials, there's not enough patients out there to power all of them, which is something people are going to have to figure out.
Interesting point there. Thank you. Replacing colonoscopy screening with a liquid biopsy blood test.
So this exists. Replace is a tough word, but Cologuard is out there. People are using it, and it's a great test.
Okay. One universal liquid biopsy panel that tells you everything about a CRC patient. Mutation status, MRD, treatment response, and prognosis. One blood draw. This is your last question.
Well, if you ask each of the companies, they all say they have it. But I think it exists in different forms and will only improve over time.
Very good. All right, Dr. Hornstein, thank you so much. Thanks so much for coming on the Moonlight Shift.
Yeah.
This was wonderful.
Thanks for having me. It's great to chat, and hopefully I didn't give too long-winded of answers.
Oh, no, you're going to come back. So,
Yeah? Okay.
this is great.
Well,
Thank
good seeing
you.